Hello! Today we're going to do some dissecting. That's a sheep kidney. You're going to get to cut that up. But before we get into cutting things up, got a couple things to cover first. Number one, notice I'm wearing safety goggles. You need to keep your safety goggles on at all times. Secondly, gloves. Both hands. Gloves. Third, an apron. I know I look lovely. You're jealous. Okay. Also, you'll notice I have my lab papers, so I know what to do. I'll follow the directions. I'll have fun, we'll be successful, and we'll be great. Okay, we have our dissection tray set up. The dissection tray is this tray that our tools and our specimens sit in. Let's go over some of these tools so you know what is what. We'll also go over the proper use of these tools. We'll start with the scissors. These are used to cut through things. They are not as sharp as the scalpel. Some things we don't need to make a clean cut, so we can use scissors. The next tool we're going to use is called the probe. The probe can be used to point and to probe into different places, different objects, to see how they go together. Next we have our scalpel. This is a knife. It is sharp. If it is sharp enough to cut the kidney, it is sharp enough to cut your skin. Be very careful with the scalpel. Always cut away from your body, never towards your body or another person. Always away. Next we have the forceps. They look like tweezers. We can use them to pick things up, to pull things back. And then finally we have dissecting pins. Little pins that we can stick into the specimen and then into the dissecting tray to help hold things in place. Now we may or may not use all of these different tools, but we have them available to us in case we need to. Next, we'll go over the actual procedure of your dissection today. First step is to just make some observations of the external anatomy. So that means you look at the kidney without cutting it, you just look at the outside. What do you see? The first instruction is to remove any of the fatty tissue on the inside of the curvature of the kidney. Right here is where the kidney curves. So the instructions are to cut away any fat that's in this area, any of the fatty tissue. I don't really see much. This might be considered the fatty tissue, but I think that's just part of one of the blood vessels. So it doesn't look like we have much to do with removing the fatty tissue. Next, you're supposed to look at the blood vessels coming out and determine which one's the renal vein and which one's the renal artery. So, coming out of this kidney, we have two sets of things. Here's this longer one for us and the shorter one. Now, if you look carefully inside the shorter one, where's the camera? You'll see that inside the shorter one there are two separate ones. And then this other one is separate from the rest. Okay? You'll learn what those are. Just know that there are two in the small one and one in the bigger one. So here we found the two blood vessels. The artery and the vein are the two in the small one. This other longer one is called the ureter. This is what carries urine from your kidney to your bladder. Okay, now it is time to get into cutting the kidney. So the instructions say cut the kidney in half the long way so you will be able to see the internal structures as shown below. So it says to make a longitudinal cut with the scalpel. Longitudinal again is the long way. You're going to cut it. So oftentimes the kidney is said to look like a bean. That's why we have kidney beans, because they look like a kidney. So when you take it so it looks like a bean, you're going to cut it like that. So I'll put it on the tray. I'm not going to cut towards myself. I'm going to cut it the long way. So that would be a cut from here down to here. You're not cutting down this way, you're cutting this way. Longitudinally. So notice I turned my kidney to its side so I can cut it and cut away from myself at the same time. I'm just going to make a cut. I'm going to try to make it as clean. I don't want to saw through this thing, I just want to cut it. Cut it and then open it. Might take a little bit of work to get it cut through. Okay, so now we have our kidney. It's cut open, and we can start to see some of the things inside of the kidney. Beautiful kidney. Now I didn't mention this before, but you might be asking what the blue and the red parts are. Well, often when we have specimens preserved to dissect, what they'll do is they'll drain the blood out of the body, because the blood easily goes bad. It will spoil. It just gets really disgusting. It's a liquid, so it won't stay where it is in the body. So what they'll do is they'll drain the blood out, and then they'll inject colored latex into the specimen. So the blue is where they injected blue latex into the veins, and the red is where they injected red latex into the arteries. So we can easily tell where the blood travels that comes going to and coming from the heart. It just helps us to see where the blood would be if it were a full and functional organ inside of the body. So we've done step number 1, which is to cut in half the long way so you will be able to see the internal structure as shown below. Okay, the picture's not very good, but this is what you'll see when you cut it open. Inside the kidney, this is number 2 now, inside the kidney, the ureter expands to form a large chamber, which is called the renal pelvis. Then the question is, what do you think is collected in this area? Remember, the ureter goes to the bladder, and the bladder is what we empty when we go to the bathroom. So the question is, what do you think collects in this area in this larger chamber? Then we're on number 3. The renal pelvis, that's this area. The renal pelvis divides into a lot of smaller compartments, which are the darker parts of the kidney tissue. The darker parts then lead into the renal pyramids. Okay, the renal pyramids are pretty hard to see, but they're going to be up in this area, in this section, right above the renal complex. So the renal pyramids are going to be just in this area right above the renal complex. And then out in this area is called the cortex of the kidney. This is where most of the filtering takes place, in the kidney. In this area of the cortex are cells called nephrons. Nephrons are cells, so you would need a microscope to see them, and they are the cells that filter out the blood to remove the waste that is excreted from our bodies through our urine. In conclusion, what we have is we have our kidney, and into the kidney from the heart comes an artery, the renal artery. It's the red that you can see right here. The blood comes from the heart and goes into the kidney. Then, as we see this red, it goes out into the cortex. And in the cortex, the nephrons, the tiny little kidney cells, they filter the blood, get out all the stuff that we need to pee out. That stuff then eventually comes here. The blood then returns back to the heart through the renal vein. That is the blue one, the blue blood vessel. So right here you can see the red blood vessel and the blue blood vessel. Blue, red. Renal artery, renal vein. Remember, this is the ureter. This goes to your bladder. This is where your urine comes from. So, the blood comes in, it gets filtered, and then the stuff that we pee out collects here with our urine. It then leaves through the ureter, goes to our bladder, and then we go to the bathroom, we urinate through our urethra. So that is a kidney. This is how you'll do your dissection. It's pretty simple, not a lot of work. This is a chance for you to get familiar with the tools, get your hands on to see real life dissections, to learn and to do.